In Chapter 2, we reviewed the basics of chemistry. Biological systems obey all the laws of chemistry, and everything that we'll do in the future in this course is based on this chemistry, and now the chemistry of macromolecules, which are putting these smaller molecules that we examined last chapter into much larger molecules. There are 4 basic types of macromolecules that form the basis of all living systems. Carbon is the framework of all biological molecules. As you know, we are a carbon-based life form. All biological molecules consist primarily of carbon, either bound to oxygen, nitrogen, sulfur, phosphate, or hydrogen. You might rearrange the letters of these to form the word sponch, S-P-O-N-C-H. That's how I've always remembered them. Carbon is unique because it can form up to 4 covalent bonds. You'll remember it has a valence of 4. That means it could connect with 4 other molecules through single covalent bonds in order to fulfill the octet rule and have 8 electrons in its outermost shell. Many molecules in biology are hydrocarbons. That is, they're carbon chains that have hydrogens attached to them. These molecules are non-polar. However, they get some unique properties from the functional groups that we might add to the side. Here on the right side, you see some of the common functional groups that we add to hydrocarbon chains to make novel molecules. Organic molecules that have the same molecular or empirical formula, for example C6H12O6, can exist in different forms. We call these different forms isomers. If there are differences in the actual structure of the carbon skeleton, we call them structural isomers. However, if there's just differences in how the groups are attached to the skeleton and how they are arranged in space, we call them stereoisomers. On the right side here, you can see an example of a specific stereoisomer called enantiomers. These stereoisomers form a mirror image. They cannot be superimposed on each other. However, they have the same arrangement of molecules around this carbon. They're said to be chiral or handed, just like our left-handed or right-handed version of the molecule you can see here on the right. They are either determined to be L isomers or D isomers, which is really whether they rotate in space to the right or the left. Now, we're not chemists, so we won't get into too much detail here, but most of our sugars are D enantiomers, and most of our amino acids are L enantiomers. Don't worry too much about the details of isomers here. We'll examine some stereoisomers and structural isomers very shortly. So then what are macromolecules? Well, they're exactly what they sound like. They're macro. They're bigger molecules than we examined in the past chapter. All the macromolecules are built from smaller molecules. 3 of the macromolecule classes that we'll examine are polymer molecules. That is, they're built up of linking monomers. Monomers are small chemical subunits. So any polymer is built up of repeating monomer small subunits, sort of like beads on a string. One of the classes of molecules that we'll be interested in today also are macromolecules. However, they are not polymer macromolecules, and that is the class of lipids. So today we'll be examining, first of all, the carbohydrates, then we'll be looking at the nucleic acids, which are DNA and RNA, and then proteins. That will cover all of our polymer macromolecules. And then the 4th category are the lipids, which include things like triglycerides and fats. There are 3 things that you want to keep track of when we look at these 4 different classes of macromolecules. First of all, what is the monomer of that? Next, what is the polymer of that macromolecule? And then what's the name of the bond called? So as we go through each of these classes of macromolecules, I will highlight each of these features. Make sure that you note what the monomer is, the polymer, and what the name of the bond is. that holds these monomers together in its polymer form. This table summarizes all of our different macromolecules. Our carbohydrates will include starches, glycogen, cellulose, and chitin. Then we'll look at the nucleic acids. The nucleic acids will include DNA and RNA. And then proteins. Proteins consist of functional proteins, which are proteins that do things like enzymes. And then structural proteins that are things like support structure. Hair and skin has a lot of protein in it. And then the final class we'll look at is lipids. Lipids have a number of different functions. They include fats, which are great storage molecules, phospholipids, which we'll see in membranes. We'll see also prostaglandins, steroids, and terpenes. So we have 4 different classes of macromolecules that make up the components of living things. And each of those is based on a carbon skeleton or hydrocarbon skeleton that has certain functional group modifications to it. So now let's examine how these monomers are held together as polymers. You can see here this circle represents a monomer of any kind, and another monomer, and a third monomer right here. These monomers are linked together by covalent bonds, which result from removing a hydrogen and a hydroxide group. Of course, this generates water. We call this dehydration synthesis. Synthesis means joining together. Dehydration, well, we're losing a water molecule. So through dehydration synthesis, we're allowed to form large molecules, and the monomers are joined together to form polymers. Now, the reverse of this is called hydrolysis or hydrolysis, to break a water molecule. So in order to break apart these 3 monomers from their polymer form, we squeeze in some water and we break it apart, sticking a hydrogen on one side and a hydroxide group on the other side. And now we have hydrolysis, or the breakdown of large molecules by the addition of water. So these dehydration synthesis and hydrolysis are the means by which we covalently bond monomers together to form polymers and then break apart polymers into monomers. So hydrolysis to break apart water in order to break apart a molecule, and dehydration synthesis is to dehydrate the molecule by pulling water out and thus sticking 2 components together. So each of the macromolecules that we'll examine in the future of this chapter have monomers which are bound together by dehydration synthesis, and they can be cleaved and broken apart by the process of hydrolysis. Let's begin by looking at carbohydrates. Carbohydrates are our sugars. They have a 1:2:1 ratio of carbon, hydrogen, and oxygen, with an empirical formula of CH02o to the N power. This N implies that it can be any number of times the CH2O, so C6H12O6. In that case, the N would equal 6. There are many carbon-hydrogen covalent bonds, and these hold lots and lots of energy. So carbohydrates are a great energy storage molecule. Some examples are our sugars, starches, and glucose. When we break apart these carbon-hydrogen bonds, energy is released. We'll examine that in the process of cellular respiration later on in the semester. Let's begin by looking at monosaccharides. Monosaccharides are the simplest of the carbohydrate molecules. They are 6-carbon sugars that play very important roles in biology, the most common of which is going to be glucose. We'll spend a lot of time looking at the structure of glucose and how it's metabolized in order to release energy in the form of ATP. Glucose has the formula C6H12O6, and it can be drawn in a number of different ways. We can have our 6 carbons straight down the center, as you see here. And in water, spontaneously, this chain of carbons will form a ring. And the ring can form in one of two different ways. It can either fold around this way, so this carbon ends up linking to this carbon, or it could fold the other way, where this carbon ends up linking to the other carbon. This leads us into two different forms of glucose. Our alpha glucose, which is the kind that we can metabolize, and then beta glucose, which is the other form of glucose when the chain folds the opposite direction. You'll notice that this also is bonded together by dehydration synthesis. We simply remove two H's and an O, and we have one ring structure of glucose. Now, a few slides ago, I discussed isomers. Fructose is a structural isomer of glucose. That is, it has the same formula, but you can notice that fructose definitely has a different arrangement of atoms than we see in glucose. Galactose is a stereoisomer of glucose. That is, it has all the same components, but you'll notice here in this middle portion that the hydrogens and hydroxide groups are on opposite sides of the carbon chain. Perhaps this helps you understand the difference between structural and stereoisomers. Enzymes act on different sugars, can distinguish from structural and stereoisomers of this basic 6-carbon skeleton. So now let's take a moment to look at disaccharides. Disaccharides are simply monosaccharides linked together by dehydration synthesis. We've examined a 6-carbon monosaccharide. However, some of our sugars will be 5-carbon sugars. For example, alpha glucose linked to fructose through dehydration synthesis, removal of one molecule of water, will form the disaccharide sucrose. Disaccharides can be used for either sugar transport or energy storage. Examples of disaccharides are sucrose, lactose, and maltose. Lactose is a sugar that we find in milk and technically is undigestible in the adult system, although some people do contain a mutation in which they maintain the enzymes that are able to break lactose apart into its monomers. However, in most humans, lactose needs to be digested by the bacteria E. coli that we find in our large intestines, and the byproduct of this is gas and sometimes a lot of extra water. So for most adults, lactose is actually undigestible, and that's why we see the gaseous bloating and such associated with the consumption of milk. For some people, the symptoms are much more robust, whereas others, it may simply be gassiness. Polysaccharides are anything more than a disaccharide. Long chains of monosaccharides linked together through dehydration synthesis compose energy storage molecules. Plants use starch, and animals use glycogen as their carbohydrate energy storage molecules. However, plants will use cellulose for their structural support, and arthropods and fungi use another polysaccharide called chitin. So let's take a moment here to look at the difference between cellulose and glycogen. They appear very similar. After all, they are a long string of glucose molecules. However, the ones that we see in starch and glycogen are of the alpha glucose form, and they link together and form bonds that look like this. We have the enzymes to break apart both glycogen as well as starch. However, when we look at cellulose that we see in the structural support of plants, they are linked by beta-glycosidic linkages. So they're linking together the beta form of glucose, which forms a different linkage, the beta-1,4 glycosidic linkage. For us, this linkage is undigestible because we do not possess the enzymes that can break these beta-1,4 linkages. So this answers the question of why we can't simply live by eating grass. If, after all, cellulose is just a string of glucose molecules, like glycogen, then why can we not live on grass like cows do? And it all resides in this alpha glycosidic linkage formed from the stringing together of alpha glucose molecules, or whether we're using beta glucose, where the orientation of the bonds is in the opposite direction and we cannot break them apart. How do cows then eat grass? They don't have the enzyme either, but they do have a host of bacteria inside of their guts that are able to break these beta-1,4 linkages, and so they can obtain nutrients from the glucose that's contained in cellulose. Same thing goes for chitin. You've seen chitin in the shells of lobsters and crabs. Spiders also have chitin, and fungi have chitin. Again, these are beta glucose linkages using beta glucose, and thus we cannot digest them fully in order to obtain all the nutrients that are stored in these molecules. So basically, what this means is paper and pasta are both made of strings of glucose. However, you know pretty well we couldn't just eat a piece of paper and gain any nutrition from it. Pasta, on the other hand, great. But we could burn paper, and as we'll explore later, burning paper in order to release heat and light is a very similar process to what we do with our pasta, where we metabolize it much more slowly and obtain energy. Rather than heat and light, we obtain energy in the form of ATP. We'll spend some time looking at that in more detail later.